Hello students, in previous part we were starting to discuss about the cell organelles. We have discussed cell membrane or plasma membrane. In this section we will discuss on mitochondria and Golgi bodies. Mitochondria. Mitochondria, first it was discovered or observed by Kolliker in flight muscle of an insect. Firstly, mitochondria was observed by Kolliker in flight muscles of an insect. The word, firstly the term word was given by Fela. The first word was given Fela by the Fleming. The term Fela was given by Fleming, and after that the term bioplast was given by Altman. But the term mitochondria, which was proposed by Benda, the term mitochondria was given by Benda. Mitochondria observed by Kolliker, but the term mitochondria which was proposed by Benda. Mitochondria is very small inside. It has only diameter. The diameter of mitochondria is 0.2 to 1.0 micron meter. Its average is 0.5 micron meter. This is the average. And length of mitochondria is 1.0 to 4.1 micron meter. So we can say that it is very small in size. We cannot see the microscope, we cannot see the mitochondria under microscope without stain. Firstly, we have to stain the mitochondria with Janus green, and then we can observe in under microscope. The mitochondria cannot be observed under microscope without stain. The stain with the help of mitochondria is colored, that is Janus green. After stain the mitochondria we can see with the help of microscope. The shape of mitochondria is just like as a sausage like. The shape of mitochondria is just like as a sausage, hot dog like structures. Mitochondria is a double membrane covered layer. It is a double membrane covering organelles. The outer membrane, which is smooth and it contains less protein. The outer membrane, which is smooth and has 60% of protein and made up of 60% of protein and 40% of lipid. So the outer membrane acts as a semi-permeable membrane. Semi-permeable membrane. The inner membrane, which is found in folding, it is found in folding, and this folding structure single is known as crista, and there are two or more, then that is called cristae. Folding structure which is called cristae or crista. Singular is crista and plural cristae. Which contains 80% of protein and 20% of lipid. Thus, the inner membrane acts as a selectively permeable membrane. Selectively permeable membrane. Outer membrane acts as semi-permeable, and the inner membrane is act as a selectively permeable membrane. How is the structure of the mitochondria present? This is the structure of mitochondria, which is oval in shape, or we can say cylindrical in shape. This is the outer membrane. Outer membrane is smooth and it has components protein and lipid. Less protein. It contains less protein rather than the innermost. The inner membrane, which is present in a folding structure, which contains this like structures. There are many folding structures. This is the inner membrane, and inner membrane act as selectively permeable membrane, and outer membrane act as a semi-permeable membrane. Inner membrane is rich of protein and which is present in folding structure. This folding structure is called, singular is called crista, and there are many more, so it is called cristae. This folding structure is filled with a fluid that is called matrix. It contains fluid. This is called matrix. Matrix contain many enzymes which help in respiration of cell. Firstly, matrix contain enzyme which help in respiration. This is the first function. Second one is it contains divalent ions. Divalent ions such like as magnesium ions, divalent, manganese and ferrous. Just in inner membrane, it contains a circular DNA. Mitochondria contains circular DNA. Which contain mostly the nitrogen base cytosine bonded with a triple hydrogen bond with guanine. In circular DNA of mitochondria contains maximum number of nitrogen base, guanine and cytosine. So, if it gains a small high temperature due to this nitrogen base triple hydrogen bond, they easily break down. So we can say that these are denaturation in high temperature. In high temperature, the bond of hydrogen bond with cytosine and guanine, they may be ruptures. So that is called denaturations. There are many. Ribosomes are also present in the matrix. These are the ribosomes. Ribosomes in mitochondria present 70S, whether the organism is eukaryote or prokaryote. it has an exceptional case. The mitochondria contain ribosome 70S. Firstly, it is observed that mitochondria is a prokaryotic cell, but then after that discovery, it is observed that it contains my ribosome that contains 70S ribosomes. Clear? There are two cavities are produced. Which one? The cavity which is produced between the outer membrane and inner membrane. This is called outer compartment. Outer compartment. And the cavity which is present just inside the inner membrane. This cavity which is filled with the fluid. This is called inner compartment. Inner compartment. The mitochondria, number of mitochondria may be present in different cell. First, it may be present in one, such like as chlorella or chlamydomonas, a single unicellular organism which contains only number of one mitochondria. And there are many mitochondria. The number of mitochondria present in cell depends on the function, how much functions the cell performs. The number of mitochondria may be different. This is due to the functions performed by the cell. Performed by the cell. It may be present in one number of mitochondria. One mitochondria in unicellular organism, such like as chlamydomonas. Or chlorella. There is only single mitochondria. In flight muscle of insect, there are many mitochondria. The largest number of mitochondria may present in the flight muscle of insect. That may be 5 lakhs. 5 lakhs mitochondria present in flight muscle in insects. And there are nearly 50,000 mitochondria may present in giant amoeba. So we can say that there are different number of mitochondria may present in cell. These are the basis of the function which is performed by cell. Giant amoeba, that is called chaos. These are the mitochondria. Mitochondria is also called powerhouse of the cell. Powerhouse of the cell. Because the energy which is provided to the cell is through mitochondria. This is due to there are some ATPase enzyme may present at the folding of cristae. There are many particles may present. These particles are called oxysomes as well as particles of fernandins. These are called oxysomes. Or we can say that particles of fernandins. This oxysome contains 3 parts. There are many folding structures, cristae, and at the surface of cristae, these particles of fernandins are also present. Those are called oxysomes. And the parts of oxysome is just like this one. Oxysome. An oxysome contains 3 parts. Base, this is the base attachment, and this is the stalk. and upper side contains head. Head contains two types of protein, F0 and F1. F0 and F1. There are two types of proteins are present. These proteins act as ATPase enzyme, which help to convert ADP into ATP. The enzyme which convert ADP, adenosine diphosphate molecule, into ATP, adenosine triphosphate molecule. Those proteins are contained F0 and F1. They are found in head of the oxysomes. This process, conversion of ADP into ATP, is called photophosphorylation. Photophosphorylation, which produce energy. So energy is synthesized by the oxysomes. Oxysomes contain three parts: base, stalk and head. This is the function of mitochondria. And next part is Golgi body. Golgi apparatus or Golgi body. It was discovered by Camillo Golgi in 1898. Golgi bodies or Golgi apparatus. It was firstly discovered by Camillo Golgi in 1898. Golgi bodies are present in animal cell as well as in plant cell near to the nucleus. They are found near to the nucleus. The main function of Golgi body is packaging the material and transport in from one place to another place. The function: packaging the material and help and transport from one place to another place. Golgi body also synthesize lysosome. The lysosome also synthesized by Golgi body. A Golgi body consists four parts. There are four parts of Golgi body. Which are those four parts? Cisternae, tubules, vesicles and Golgi vacuoles. Golgi vacuoles. These are the four parts of Golgi bodies. Firstly, we draw the diagram of Golgi body. Golgi body consists two parts, two surface. One is concave and other one is convex. There are many plain sac-like structures which are wider at the end. There are 4 to 8 such sac-like parallel membrane arrangement in Golgi body. The end of all the sac or tubular structure are just wider than the other membrane. These are this one like structures. These parallel arrangement, one side that is concave and other one is the convex. The convex surface remains toward the plasma membrane. This is the convex, remains toward the plasma membrane, nucleus membrane, and the concave surface remains toward the plasma membrane. These parallel arrangement of sac-like structure is called cisternae. The parallel sac-like arrangement is called cisternae. These cisternae are attached internally with a tubes like structures. This one like. These are called tubules. So tubules, the tubes which connect one cistern to another, that is called tubules. Which connect the cisternae, that is called tubes. Golgi vacuoles at the corner of cisternae. There are large vacuoles. These are called Golgi vacuoles. At the edges of cisternae. The swollen, or we can say the wider part, is called Golgi vacuoles. These Golgi vacuoles breaks into smaller and larger piece. Golgi vacuoles, if they break down into larger piece, these larger piece remains here, just towards the upper side, and in smaller they remains lower side. These smaller Golgi vacuoles breakdown pieces are called vesicles. These are called vesicles. And larger, which some of them convert into lysosome. Lysosome, which contains some enzyme, and if there is malfunction happened, disturbance occurred in the metabolism process, the lysosome break down its own cell, so they are called suicidal bags. There are two surfaces of a Golgi body. One towards the upper side, this is called maturation phase. Maturation phase or maturation cyst. Towards the maturation cyst, some lysosome and large vacuoles are present. And the other surface, this is called formative cyst. Formative cyst. And it contains vesicles. This is the structure of Golgi body. Golgi body was discovered by Camillo Golgi in 1898. The main function of Golgi body is to help to package all the material and help in transport from one place to another place through the ER. The Golgi body contains 4 parts: cisternae, tubules, vesicles, and Golgi vacuoles. Cisternae, these are parallel arrangement of 4 to 8 cells. These parallel arrangement of cells, sac-like structure cell, is called cisternae. At the edges, the cisternae which is swollen or wider, this is called Golgi vacuoles. Each cistern attached with the other, some tube-like structure, those are called tubules. Clear? The Golgi vacuoles breaks down into smaller or larger piece. So larger piece, which make some lysosomes, and they remain for the maturation phases. And the smaller piece, small vacuoles, those remains towards the formative cysts, lower side, and these are called vesicles. This is the Golgi body. Next part we will discuss on some other cell organelles.